Dear students, I am going to discuss a very important topic, that is soil fertility evaluation. Soil fertility evaluation. Soil fertility evaluation. What is soil fertility evaluation? Diagnosis of nutrient status of the soil by using different techniques and methods, that is known as soil fertility evaluation. Methods of soil fertility evaluation. There are various diagnostic techniques which are commonly used to evaluate the fertility of soil. They are soil testing, plant analysis, biological tests, and visual symptoms and plant. These are the methods of soil fertility evaluation: soil testing, plant analysis, biological tests, visual symptoms in plants. Today I will cover only soil testing. Soil testing is a chemical method for estimating the nutrient status or nutrient supplying power of the soil. It gives a measure of nutrient availability to the plants. Whereas plant analysis gives actual removal of nutrient from the soil. Soil and plant analysis is an important tools of the recommendation for the application of fertilizer to the crops. It's play a key role in today's modern and intensive agriculture production system. Soil testing is helpful in better understanding of the soil to enhance or to increase the crop production and obtaining the sustainable yield. What are the objectives of soil testing? Objectives of soil testing. First one is to evaluate the fertility status of the soil by measuring the nutrient status in soil. Next one is to recommend or to prescribe the soil amendments like gypsum, lime and fertilizer for each crop. And next one is the to know the salinity, alkalinity and acidity problem of the soil. Salinity, alkalinity and acidity problem of the soil. And next one is the to know about the morphology, genesis and classification of the soil. And next one is the to prepare a fertility map of an area like village, districts and states. This is the objectives of soil testing. Collection of soil samples from field. How we can collect the soil samples from the field? For collecting the soil samples from the field, some materials are required. Materials which are required to collect the soil samples from the field, that is spade or auger. Second one is the khurpi. Core sampler. Sampling bag. And next, plastic tray or container. These are the materials which are required to collect the samples. Before collecting the soil samples, some precautions are needed before collecting the soil sample from the field. Precautions which are required to collect the soil samples. Before collecting the soil sample, some precautions are required to collect the soil sample. First one is to collect the soil sample from fallow periods. Collect the soil samples from fallow periods. Next, in standing crop, collect the soil samples between the rows. Collect the soil samples between the rows. in standing crop, collect the soil sample between the rows. Next one is the to collect the soil samples from the field, follow the zigzag pattern for the sample. Follow the zigzag pattern for sample. What is zigzag? Suppose this is your field. You collect the sample from here, here, here. This is the zigzag sampling pattern you should follow. Next, fields which are similar in appearance, production and past management practices. Field can be grouped in single sample units. Field should be divided in single sampling units on the basis of their similarities. Field could be grouped in single sampling unit. Next point is collect the sample from the fields which are differ in color, slope, drainage, and past management practices and cropping system. Next one is the avoid sampling from dead furrows, area near the main one, and trees irrigation channel, manure heap, etc. Next point is collect the soil sample to grow the shallow rooted crop up to the depth. For shallow rooted crop, collect the samples up to the depth of 15 centimeter. For deep rooted crop, collect the samples up to the depth of 22 centimeter. For plantation, and orchard, plantation crops and orchard, take the samples up to the depth of 90 centimeter. It can be done 0 to 30 centimeter, 3 sampling from 0 to 30 centimeter, 30 to 60 centimeter, and 60 to 90 centimeter. These are the precautions you should follow before the sampling from the field. Procedure for sampling. Procedure for sampling. First one is the divide the field into the different homogeneous units based on the visual observation and farmer's experience. First of all, you should divide the field in different homogeneous units based on visual observations and farmer's experience. Second one is the remove the surface litter at the sampling spots. Remove the sampling, remove the surface litter from sampling spots. Next point is drive the auger to a plow depth of up to 15 centimeter and draw the sand. Use auger up to the 15 centimeter depth and draw the sand. Next one is the collect at least 15 samples from each sample units and place into a clean container or tray or bucket. Next point is, if agar is not available, make a V-shaped cut up to the 15 centimeter depth on the sampling spots and collect the sample. This can be understood in this way. If agar is not available, make a V-shaped cut to a depth of 15 centimeter depth in the sampling spot using spade. If agar is not available, make a V-shaped, English letter V-shaped cut to a depth of 15 centimeter up to 15 centimeter depth of 15 centimeter by using spade. Suppose this is your field, you cut the soil in V shape. Cut the soil in V shape. This 15 centimeter depth with the help of a spade, and remove the 2.5 centimeter slices of soil. Cut the slices of soil 2 point from the exposed V cut shaped face, exposed face of V shaped cut. You cut the slice of 2.5 centimeter from the exposed face of the V shaped cut and collect the soil samples into the clean bucket or container. Next, mix the samples thoroughly. Next point is, mix the samples thoroughly and remove the crop roots, pebbles, gravels and stones from the samples. Next point is quartering is done. Quartering is done. Quartering is done by thoroughly mix soil sample and divide into the 4 equal parts. It can be, and the 2 opposite quarter of soil is discarded and remaining 2 are remixed, and procedure is repeated until the 500 gram sample, soil sample is obtained. How we can done the quartering of samples? Thoroughly mix the samples. You can understand in this way. Quartering the samples. Now first, second, third and fourth quarters of soil sample, it mix well and dividing into 4 equal part, and first 2 opposite quarter of soil is discarded. This and remaining 2 part of soil is taken and remixed, and procedure is repeated until the 500 gram of soil sample. This is the procedure of quartering of the sample. Next, collect the sample in clean cloths or in clean cloths or in polythene bag. Next point is label the bag. Labeling the bag with some information, like name of farmer, location of the sample, third one is the sample number, name of previous crop, is grown, and name of present crop which will be grown in next session. Date of sampling, and name of sample, sampler. name option. These are the, during the leveling, these are the information should be write on the during the leveling of the samples, and these samples dispatched to the soil testing laboratory. So today's I have covered about soil testing, and I will continue this lecture in next part. Thank you.